Hello, everybody. My name is S.Wang. I'm a program director for the Brain Initiative. I'm going to introduce Brain-funded electromagnetic recording projects. First of all, a brief history of Brain support of such technology development. The Brain Initiative made the first funding opportunity announcement for non-invasive brain functional imaging technology development in 2014 as a 3-year preparation grant mechanism. In 2016, we published the first full-scale development funding opportunity announcement and made the first award in electromagnetic recording in 2019. Since then, we have funded 2 projects in this space, one in magnetoencephalography and the other in acousto-electric imaging. Materials of the rest of my presentation come from these 2 resources. In February 2021, the Brain Initiative held a dissemination of a non-invasive imaging technology workshop. We invited all awardees to present their technology. The recorded video can be found at this website. You can also find the detail of this project at NIH Reporter website. So the first project we funded is led by Dr. Peter Schwint from Sandia National Lab. He proposed to develop optically pumped magnetometer to measure the weak magnetic field generated by neuronal current from outside of the human skull. Conventionally, this type of measurement is performed by low temperature superconducting quantum interference device, or so-called SQUID. It requires a large dewar to contain liquid helium to cool the device to sufficient low temperature. As a result, there is a large standoff distance between cortical surface and the sensor, which reduces the signal intensity. So in this project, Dr. Schwint proposed to vaporize rubidium and shine a laser through it. As the laser travels through the rubidium vapor, the amount of polarization change becomes dependent on the magnitude of a magnetic field. In this way, one can measure the very weak magnetic fields without using the low temperature SQUID device. So that can reduce the standoff distance by nearly half, which significantly improves MEG signal spatial resolution and information contents that is able to access. Also, it's possible to custom design the housing of these OPM detectors by using 3D printing technology, which enable imaging human subject in naturalistic behavior. The second project we funded is led by Dr. Russell Vitti from the University of Arizona. He proposed to use ultrasound waves to facilitate the measurement of very weak electric signal outside the human skull. Conventionally, this is performed using electroencephalography technology, or so-called EEG. Because the human skull is electrically lossy, the spatial resolution of EEG is limited to about 2 centimeters. So in this project, Dr. Vitti proposed to sonify a tiny piece of brain tissue by using ultrasound wave. In response to acoustic pressure, the brain tissue expands or contracts, and its electrical impedance changes accordingly. So when the neuronal current flowing through this tiny tissue, the voltage detected outside the brain become modulated by ultrasound wave. In this way, this technology can significantly improve the spatial resolution from a centimeter scale down to a few millimeters, depending on the ultrasound frequency. Because it does not require solving an inverse problem to find the source of the electric signal, it is capable of imaging deep brain neuronal currents, which is unimaginable by conventional EEG technology. So with this, I conclude my presentation. Thank you.